Hello everyone, my name is Mathias Witte Paz, and today I will be presenting our work on TSS-Captur, a transcription start-site-based characterization pipeline for transcribed but unclassified prokaryotic RNA transcripts. I know, long title, but the keywords you would like to keep in mind for this talk are transcription start-site, so TSS, and characterization pipeline. In the recent years, we've seen the development of specialized RNA-seq protocols for the prokaryotic transcriptome. With dRNA-seq and capable-seq, we've allowed to enrich such RNA-seq libraries to get some 5' enriched libraries, which we can then use with computational methods to identify the exact base of a TSS. One of these computational methods is, for example, TSS Predator. Then, if we take a look at the distribution of the TSS, we see that the majority of them are found upstream of annotated genes. However, we have also identified signals that are nowhere close to any annotated gene. These TSS are called orphan TSS. And this arises a question, is it possible that we've identified new genes using transcriptomic data? And if so, are we able to characterize them even further? For example, are there non-coding RNA genes or coding genes? Are we able to identify a termination site downstream of this TSS? Or if we take a look at the upstream region of this TSS, are we able to identify any enriched regulation motif that might provide insight into the transcription of this transcript? There are already tools that are trying to characterize such unknown transcripts. However, they still have some disadvantages. For example, Apero and Burhunter, they are basing their predictions just on RNA-seq when it has been proven that such 5' enriched methods provide a more accurate position for the TSS. Another tool is Anogesic. This one is basing the predictions on based on dRNA-seq and is also providing an extensive characterization of the transcripts. However, it still has some disadvantages. For example, the classification of the transcripts is based on a simpler approach. It's just accounting for the intrinsic termination for the transcripts, even though there are further termination methods for prokaryotes. And it's missing one key part, which is providing the users a way of interactively exploring the results. These points we would like to tackle with TSS-Captur. TSS-Captur is a Nextflow-based pipeline which is going to characterize the transcripts on different levels. After the extraction, the transcripts are then classified as coding or non-coding RNA genes. Then we're trying to predict a termination site. And for those that are classified as non-coding RNA genes, we are analyzing the possible secondary structure. With using the whole set of putative promoter regions, we're running an enrichment analysis on their motifs to identify enriched motifs that can provide insight into the regulation of these transcripts. At the end, all of the results are then combined into one interactive web interface for the users to explore the data even further. Now let's go deeper into each steps of TSS-Captur. At the beginning, we're extracting the transcripts starting from the TSS, and we're avoiding any collision with any other TSS or any other annotated gene. Afterwards, we're classifying the transcripts using two different tools. qRNA for the detection of structural changes in a pairs alignment to classify the transcripts as RNA or coding, and CINIT, which is an SBM-based classifier which is trained on eukaryotes, since we haven't found any other tool that has the same purpose but has been trained on prokaryotic data. Afterwards, we are predicting for each TSS a putative termination site using two different tools. Transform-HB accounts for the intrinsic termination in prokaryotes, while RAW-Predict accounts for the RAW-dependent termination in these transcripts. For genes that have been classified as non-coding RNA genes, we're using RNA-fold to find the most stable structure with respect to the minimum free energy. And using MEME, we're analyzing the whole set of putative promoter regions to identify motifs that are enriched within the whole set of sequences. This motif can be then afterwards compared by the user to see if they have some resemblance to known regulation motifs, for example, the Primno or Tata box. All of the results are then summarized into one single interface, a web interface, where the user can explore this data even further, but I'm going to show an example in a few minutes. To show you an example, I will be using a dataset that was produced for Streptomyces cellicolor using dRNA-seq, and then we have pre-process using TSS Predator. To see which of these genes are actually experimentally confirmed, we've been using a set that was confirmed by Fockenhuber and colleagues, and this showed an intersection of 23 orphan TSS. So this is the overview of the results of TSS-Captur. And so you can see here we have the 23 TSS, and the user also gets an overview that 5 of them were classified as coding and 18 were classified as non-coding. We also have a table where the users can sort depending on what they would like, for example, the classes shown here, or with respect to the motif that might have been found in the upstream region of each transcript. Users are also able to search for specific positions, and for example, this transcript here is classified as a non-coding RNA, and hence we can directly visualize the non-coding secondary structure that was produced by an RNA fold. We are also showing detailed views on each of the single steps of TSS-Captur, for example, the classification or also the termination allocation. And we're providing a direct link to the results that were analyzed with MEME, so the users can have everything within one interface and hence have an easier linkage of the results. With this, I would like to end my talk where I presented TSS-Captur, which is a pipeline which is facilitating the characterization of this transcript starting at orphan TSS positions, but also providing a way of exploring this data even further. One of the next steps we would like to continue development is replacing CINET with a classifier that has been actually trained on prokaryotic data. So if you'd like to talk with me about this, you can come over poster or contact me via Twitter. Otherwise, I would like to thank our funders and also you for your attention, and please contact me anytime for your questions. Thank you very much.